Dr. Gary Greenberg. I first met Gary. He gave an interesting talk on an unusual microscope that was like nothing I had ever seen. And I said, gee, Gary, can you build a differential interference contrast version? He goes, well, that would be cool. And I said, great, and, why don't you come over? And he kindly brought all of his equipment over to the labs. What we do here is remote sensing, and we look at things trillions or quadrillions of miles away. But what Gary does is also remote sensing. Remote sensing, it's not the psychic thing, right? Remote sensing is when you measure something at a distance. I could look outside at the grass and go, it's green, it's absorbing this color of light, and it's got chlorophyll A, and without touching it, we can measure what is there. So Gary is in my opinion, the best microscopist in the world. We have a project together where we are analyzing moon dust, where he's doing interesting 3D pictures of the moon dust, and we're looking at the 3D chemistry. One of my interests is in situ resource utilization. So looking at moon dust so you can make concrete or get water or things like this. So we've been very happy to have Gary here for now, what, a couple of years has it been? Okay, so Gary is an unusual, how many here have looked through a microscope? Okay, and so when you look, you see that there's a little bit in focus and then a lot of it's blurry, right? And so it's a fast optic. It's the light going up into the lens goes at an angle like that, so you don't get a lot of depth of field. And so Gary's worked out interesting techniques to get a long depth of field, but also he does this amazing 3D microscopy, and humans see 3D by looking mainly by you have a slightly different angle. If you look at one eye with the other, there's a slightly different angle, or the things that your retina, your brain knows how to make 3D information. Okay, well, with that, we've tap danced long enough to actually get the presentation going, keep you entertained. So without further ado from me, let's welcome Dr. Gary Greenberg. Sure.
That's a challenge. Ethel Merman, she could belt it out. I didn't know you're old enough to even know who Ethel Merman is. Gee. Man. Well, thank you all for coming tonight. I really appreciate it. And what I'm going to talk about tonight is my favorite subject, the extraordinary nature of ordinary things. I've been looking through microscopes for a lot of years, and I never cease to be amazed at how incredible just ordinary things are when you look at them through a microscope. So that's some of what we're going to look at tonight, but what I want to really focus on is how has science changed the way that we see light, time, and space? Because over the years, science has had an incredible influence in the way we see the world around us. I want to start off with one of my favorite Renaissance paintings by Hans Holbein. It's called The Ambassadors. And in this painting, there's a couple of ambassadors standing there, and there's two levels on this table. The bottom level shows the worldly things like music and a globe, and the top level shows more of the spiritual life in the spiritual world, such as things in the stars and the heavens. And the amazing thing you see about this incredibly well painted picture is this strange thing right here. It looks like a UFO. It's just like, was there a mistake in the picture? Is there some damage to it? Well, it turns out this was an interesting statement by Hans Holbein. If you look at this picture tilted, then you begin to see what this is going to be to look at. If you tilt it really a lot, this is what it looks like. It's a human skull. So there's a human skull at this incredibly oblique angle stretched out across the bottom of this painting. So what Hans Holbein was trying to say is we humans see the world from a very limited point of view. And that's actually what we do as human beings. We have the five senses. Dogs see the world very differently, and ants see the world very differently, and elephants see the world very differently. So we see the world as human beings. And he was trying to make the statement, we really are limited in the way we can see things. We don't see things as they really, truly are. In fact, nobody can see things as they really, truly are. So let's start with one of the first questions that man had asked. What is our place in the universe? So we use our point of view. Our point of view determines what we see. And our point of view as early hominids, and we wake up from the cave and we see the stars and the sun going around. So it looks like that we are on a fixed Earth, and the stars and the sky and the sun is going around. It doesn't look like, actually, we're spinning. Let's see, we got a 24,000 mile diameter. It takes 24 hours. So we're spinning about 1,000 miles an hour. It doesn't look like that, but we actually are. So the first approximation at our place in the universe is that the Earth is the center of the universe and everything revolves around it. But that, let me just get a little picture here. So, and the reason we do that is because our senses are used to perceive the world around us. So religiously, and in terms of all of the legends go, the Earth is fixed and is at the center of the universe. Well, what's interesting is this started to change in the Renaissance time. And in the Renaissance time, Copernicus came along and said, you know what? It works out a lot better if the sun, if we're going around the sun, the mathematics works way better. Now, it turned out that a guy named Aristarchus figured this out, didn't figure it out. He proposed this a 1,000 years earlier. But it wasn't taken seriously because nobody would take seriously the idea that the Earth was not at the center of the universe. But Copernicus was a really good mathematician, and he showed that the mathematics really works out. So it became a much more compelling thought that the Earth is not at the center of the universe. And what really started to clinch this was shortly after the telescope was invented, and it was 1600 in Holland, and a dozen or 2 years later, Galileo got a hold of the telescope. And originally, telescopes were not used to look at the stars. They were used, the Navy was interested in them, and they would look at things like the enemy and use it for navigation. Galileo was the first to really turn the telescope up to the heavens and see what it looked like. And he started looking at Jupiter. And he made these little drawings every night to look at Jupiter. He noticed that there were little dots of light around Jupiter that kept moving. And he started plotting them. So on one night, they were there, and there. The next night, there were 3 in a row. And on some nights, for example, on night 15, there were 4 in a row. And then in the middle of the night, the one got occluded by the, one was eclipsed by Jupiter. And on night 19, there were 3, and then later in that night, another one popped out. So he proposed that these were moons of Jupiter, and that these moons were literally traveling around Jupiter. All of a sudden, there's scientific evidence that the Earth is not the center of everything. Everything does not revolve around the Earth. It was pretty compelling information, and he got in a lot of trouble for proposing this idea, because the church had a vested interest in saying that the Earth is the fixed center of the universe. And in fact, this is the height of the Inquisition, and he was put under trial by the Inquisition. And when he was on trial, he tried to make a case that didn't bear well for him, because he didn't do well with the Inquisition. But he said, you know, God wrote 2 books. God wrote the book of Scriptures, and God wrote the book of nature. And if they should conflict, the book of nature should win out. The church didn't like that idea. That's what really got him into trouble. Not so much that he found the moons of Jupiter, but that he should make that kind of a statement. So let's fast forward a couple hundred years to Edwin Hubble. And now all of a sudden telescopes are getting big. And we've built a 100-inch telescope in Mount Palomar. And now astronomers used to look at things called nebula. And they were kind of fuzzy places in the sky. So you could see stars, and stars would make a pinpoint image on a film. But there were some things that were nebulous, and they sort of were kind of fuzzy. And now when Hubble started looking at this using the 100-inch telescope, it turned out that those nebulae were entire galaxies. Nobody ever thought that there was something outside of our local galaxy, the Milky Way. And he showed and proved that these nebulae were actually not in our galaxy. They were way far away. And they were consisted of actually billions of stars, hundreds of billions of stars. So what he did also, he wanted to find out what these nebulae were made of. And what astronomers would do in those days, and still do, is they look at the starlight, and they spread out the starlight either with a grating or with a prism, and they see that when they spread it out, that depending on what the starlight goes through or what material is creating the starlight, you get either absorption lines or emission lines at exact places in the spectrum. So for example, this is characteristically hydrogen, and this is sodium, and this is calcium, these absorption lines. And that's how you can tell what stars are made of. If you do it in a lab, you can use these chemicals, and you see every time it comes out that way. Well, what Hubble found was something very interesting. When he looked at the spectrum of these distant nebula, these galaxies, which he thought were galaxies, he noticed that all the lines, this is the hydrogen line, everything got shifted to the red. They weren't where they supposed. The lines were all there in the proper order and the proper percentage, but they're all shifted toward the red. It's called a red shift. So we're all familiar with this idea in sound. You can be on a racetrack and you hear the car coming toward you. So when it goes away, it's a low sound. When it comes toward you, it's a high sound. This happens with light as well. So the frequency, or when something is going away from you, these lines would be shifted to the red. If a galaxy or a star is coming toward you, the lines would be shifted to the blue. So he proposed that these galaxies he was looking at were moving away from us. And the interesting thing is the farther away these galaxies were, the greater the redshift. So they were moving away even faster. So he proposed that we're in an expanding universe, that the universe is way more than just the Milky Way. There are thousands, actually millions of other galaxies, and they're all expanding away from each other, almost all of them. Some of them are coming toward us, but most of them are expanding. So now look at our view of the universe, our place in the universe. It's really quite different. Now we see we're in an expanding universe, and there's no center and there's no outer edge. It turns out when you look, if you take a little patch of black sky where there's hardly anything there, and you put the Hubble telescope to look at that little dark, tiny patch of dark sky and keep the camera open for a long time, you see hundreds and thousands of galaxies. And you're looking way back at the beginning. As you look farther and farther back, you're looking farther back in time. So when you look at the sun, you're looking at what the sun looked like 8 minutes ago. When you're looking at the closest star other than the sun, you're looking at what the star looked like 4 years ago. When you look back at these very, very distant spots in the star, you're looking at 12 billion, 13 billion years, about 12.5 billion years ago. So the interesting thing is it doesn't matter where you are in the universe. As you look farther back, it's going to be 12.5 billion years, no matter where you are. There's no center. We all are in the center. And a lot of my students say, well, where is the edge? You know, if it's an expanding universe, where's the edge of the universe? Well, if you look all the way back where you think the edge is going to be, that's the beginning. It's not the edge. This is the edge, here and now. We are at the bleeding edge of the universe, right here. So now we have a very different view of our place in the universe. When I graduated college, I saw this photograph here. This photograph really, viscerally, made me realize what the Earth is and where we are in the universe. So when we saw this picture, this is from the Apollo mission, went to the moon, went around the moon several times, and it took and then we landed on the moon, and it took pictures of the Earth rising above the moon. This really, for the first time, people who are my age would know the significance of this picture and how a simple picture like this can completely change your point of view of the world that we live in. So what I want to do is now talk about unique points of view, because we see things differently depending on our point of view. And if we can look at things from a very unique point of view, we see the world in a wonderful new kind of way. And it expands our range of perception. So I want to show you how science and technology has allowed us to look at the world in these amazing new ways. So for example, human perception is limited, as we saw in the beginning. We can't see a bullet going through an apple. But technology allows us to see that. We can't see stars exploding in front of our eyes, but with telescopes we can see them. And we can't see atoms. But with microscopes we can now begin to see atoms. So all of these things are becoming part of our perception because of science and technology. And I want to show you a little bit how that happens. So to begin with, let's talk about this little bee here. I want to talk about vision. We have a certain resolution of vision that we can see in the world. Other animals have different amounts of resolution of vision. So let's look at this little bee here. That's the eye of that bee in a microscope. And you can see the eye of the bee is made of hundreds or actually thousands of little ommatidia. They're little individual eyes. Each little eye is hooked up with a nerve cell. So what this bee sees in the world is a mosaic of about 1,000 spots of light. And that bee has two eyes, so it sees all the way around. It doesn't see in 3D, in stereo, but it sees all the way around these little dots, a couple thousand dots, and that's how it navigates the world. Now let's look at an ant. There's an ant. Look, he's got about 100 ommatidia. So he doesn't have very good resolution in his vision. And in fact, this is about 100 pixel resolution that ant has. If that ant looks at a circle or a square, the exact same receptors are going to be excited. So they'll look at the same to him. He can't even tell the difference between a circle and a square. That's how bad the resolution is of an ant. Now let's look at humans. Humans, this is the human retina, and the human retina is really good vision. We have about 6 million cones for color vision, which is in this area right here, in the center, in the fovea, and we have about 120 million rods for black and white vision. So we have some pretty darn good vision. And humans are highly visual animals because of that. But let's look at something else. Let's look at a bird. Some birds have 5 times better resolution than we have. And not only do they have 5 times better resolution, they can see the Earth's magnetic field. And they can see polarized light. So their vision is like way better than ours on all kinds of levels. So let's talk about how our vision is limited. Our vision is limited because we only see what's called visible light. So if the world is bathed in electromagnetic radiation. There's electromagnetic radiation coming from almost all matter, or much of the matter, and we're bathed in this electromagnetic radiation, and all we see is this little strip here, this little bit, we're expanding it out, and those are the colors that we see, from 400 to 700 nanometers of resolution. So it goes from red to deep blue, and we can't see anything more. But in fact, the radiation is there. There's ultraviolet radiation. This is higher wavelengths, and then finally X-rays, and the very high power ones are gamma rays. And looking at the longer wavelengths, we can't see it, but there's infrared. We're all radiating in infrared, but we don't see it. And then radio waves, radar, TV, FM, AM, these are very long wavelengths. So we only see this little tiny window of resolution. As I say, we're all radiating infrared. If you go in a dark room, completely dark, and get a thermal camera and take a picture of somebody, they're glowing. We're absolutely glowing in infrared. It's heat. In fact, if you put your hand to your face, don't touch it, you can feel the heat being radiated just by putting your hand close to your face. We are radiating this electromagnetic radiation, but it's invisible to us. But it's not invisible to everybody. Now let's look at our apparatus where we can see. So we have 3 different receptors for looking at color vision. We have a type of cone receptor that peaks at about 460 in the blue. We have one that peaks near the green, and another one that's close to it that peaks in the red. So we see red, green, and blue. And all the colors that we see are combinations of red, green, and blue. The millions of colors that we get on our computers, all combinations of red, green, and blue, because that's all we see, and we only see by these different photoreceptors. Look at this. Look at the photoreceptors of a bird. First of all, they're really evenly spaced, not like ours, and they get an entire other, they have 4 photoreceptors, and one of them is fully into the ultraviolet. So they see ultraviolet light. We can't see it at all. So they have a really incredible palette of vision, way better than we have. Now let's look at this. If you were a bird, or if you were a human, that's what this flower would look like. But if you're a bird or a bee, you can see ultraviolet light. And in ultraviolet light, that same picture has a different palette of colors. So now the bee and the bird sees a design that we can't see. If human sees a flower like this, a bird or an insect sees it like this. Human sees a flower like this, a bird or an insect sees it like this. We can't even see this pattern here, but it's there. It's there in the infrared region. And a lot of flowers are designed to give off to reflect light of infrared to attract birds and bees. So a lot of the real beauty of flowers, we don't even see it. We don't even see the colors that are there. Now, we've used this ability to see infrared and ultraviolet and all kinds of interesting wavelengths. And here's a great, interesting picture of a star. This is our closest star, the sun. And it's being, and what this is, it's an extreme ultraviolet. There are various filters were used to look at this thing in an extreme ultraviolet, at different ultraviolet wavelengths. So the different colors represent different temperatures that the sun is. So for example, the blue represents about 800,000 Kelvin degrees. The green is about 1.3 million, and the red, which is the hottest part here, that's about 2 million degrees. So now we can all of a sudden use ultraviolet information, which we couldn't even see with our eyes, but we can colorize it and use that to give us information about the world around us. So here's another example. A lot in astronomy, we use false color images. When you look at, if you look at a nebula, this is really interesting. This is an image taken with 3 different space telescopes. One of the space telescopes is the Chandra X-ray image, and that's the light blue. So it's really bright. This was an exploded star. And the X-ray image shows unbelievably high energy right in the middle, giving off a very, very high amount of X-rays, and there's a neutron star in the middle of this thing. And then another one, the Hubble Space Telescope, in green, it's looking at the visual, in the visual field that we all can see. And then there's another space telescope, the Spitzer Space Telescope, that looks at infrared. And they've combined all these images, and that's in red. So now we can look at this nebula, at this exploded star, and get much more information because we've used false color, and we use these incredible telescopes in space to give us windows to all these different forms of radiation. We also do this in biology. Using false color in biology has really changed biomedical research quite incredibly. So this is a picture I took of this is muscle cells. These are called striated muscle cells. And this here is muscle cells, and those are muscle cells from smooth muscle in your arteries. Or these are little arterioles, little tiny arteriole. You know that our veins don't pulsate, but our arteries and arterioles, they have muscle walls, and they actually contract like the heart does. And the blue here, that's all DNA. So we've figured out ways in biology to stain the biological material we're looking at with very exact stains so that everything that it looks at will tell us exactly what that material is. So the red is actin, it's a muscle protein. The green is myosin. Where green and red come together, it makes yellow because actin and myosin cohabitate together. So it looks yellow, and the blue is the DNA. We've also been able to use something called a green fluorescent protein. This is somebody isolated the gene for jellyfish protein. You know when you go to the Maui Ocean Center and you see these jellyfish and they light up fluorescently? Well, they have isolated the genes that do that, and they can combine those genes with any gene in the body so that when that gene is turned on, we see it glow in green. And they've developed blue ones and red ones and yellow ones. And so you can do this labeling and follow any gene in the body and see when it's turned on. Is it being used in health and disease? It's been a very, very powerful technique to use these false color techniques, to use our colors to expand our perception of the world. So I want to talk a little bit about time now. You know, we think of time as really, you know, sort of a fixed thing. Well, this is a painting by Dali, because at about this time, Einstein was coming up with some really interesting ideas about time. I mean, some really interesting ideas, like that time isn't as exact and as absolute as we thought. And a lot of artists started picking up on these themes. So Einstein had these ideas where he'd do thought experiments. You know, Einstein never actually was in a lab and did his experiments were always thought experiments. So I want to show you a little thing here. Imagine a thought experiment. There's a train going by, and there's a person on the embankment, and there's a person right in the middle of the train. So here's the person on the embankment, there's the person in the middle of the train. Now, at one moment, these two flashes of light, at the beginning and the end of the train, they go off simultaneously. So simultaneously, two flashes of light goes off. Let's see what the person on the embankment sees. The person on the embankment sees the two lights traveling to him arriving at exactly the same time. But let's look what happened to the person on the train that's moving. Because this person on the train is moving in this direction, it takes some amount of time for the light to reach that person. So by the time the light reaches this person, the front light hits first and the back light hits later. So all of a sudden, here's a phenomenon that we think happens simultaneously. But there's no such thing as simultaneous in the world, because everything is relative to a moving frame of reference, and that's what Einstein showed as part of his special theory of relativity. So he really changed the whole way we think about time, and time gets stretched and warped and all kinds of things. This is a great little painting by René Magritte, as sort of an homage to Einstein's. It's called Time Transfixed, talking about clocks and moving trains and stuff like that. So it was sort of a artistic takeoff on Einstein. Time also, once we got movie cameras, actually this is before movie cameras, this is from a series of stills. Leland Stanford was an industrialist in California, and he had a bet with a friend whether a horse has all of its feet off the ground at one time during its gait. And you couldn't see it because it happens too fast, and nobody could tell for sure by looking at a horse gallop, does that really happen? So he got his friend Edward Muybridge to set up a series of cameras, whole series of cameras, and have the horse trot by, and as the horse came by, the cameras were triggered. And he got a series of photographs that got put together into a movie. And in fact, right there, all the feet of the horses off the ground at one time. So it shows how we can use technology and cameras to slow time down, to analyze time in a way that we can really begin to understand stuff. Another guy, Harold Edgerton, started developing strobe lights. He figured if he could develop a strobe light that gave a really bright flash but really small amount of time, then it wouldn't matter about how fast the film is. He could freeze time in an instant by using a strobe light. So it's in a black room. The strobe light goes off for a fraction of a second, and this is a drop that went into the milk. It's a classic picture. You've all seen it. And the drop comes right back up. In fact, if you take some milk or coffee, whatever, do this tonight. You'll see. It's quite amazing. You put the drop in. You can actually see the drop. The drop bounces off the surface like a trampoline, and it comes, and the drop comes right back up. You can actually see it if you look for it. It's pretty amazing. So with these techniques, we can freeze time. We can do all kinds of interesting things with time. Now let me see if this is going to play. I want to play a little. So this is time-lapse. Time-lapse allows us to see the things that we can't possibly see otherwise. So if you just relax a little bit, a lot of time is going by, a lot more than is going by right now. And you can see things that you could never, ever otherwise see. So we've seen dandelions 1,000 times, haven't we? But we've never really seen it like that. Let me see what I'm doing here. Okay, I think, yeah, good. So I want to talk about how most of the space in this world that we live in this universe, we really don't see. Most of the space is hidden from us. And we can use telescopes and microscopes to see the space that's hidden from us. I want to show you a little bit about that today, especially with microscopes. So microscopes were invented the same time the telescopes were invented, literally exactly the same time in Holland, 1600. And no one ever expected there was a microworld. No one ever thought that there would be a microworld. So it was kind of a big surprise when Robert Hooke was the first to really adopt this in a strong way. And this is already 65 years later. He made a book called Micrographia, and he took pictures of all kinds of ordinary stuff. Fleas, bugs, flowers, just stuff that is part of the world that they all lived in. And he made this huge book out of it. Now, those days they didn't have photography, so he was a really good artist, and he could draw these things really well. And he made incredible drawings of the microworld in this book called Micrographia. So that's a flea. That's a flea. Now, nobody had any idea that their dogs and cats were filled with these things. And because they just see this little dot, right, and it's jumping around. This is what a flea looks like. And he showed that people were amazed, absolutely amazed. He introduced the microworld to human consciousness. That's a bee's eye. People had no idea that bees had these compound eyes. So he really introduced an amazing world to human consciousness that didn't exist before. Shortly thereafter, a guy named Leeuwenhoek in Holland got very impressed by micrographia and decided to make himself a little microscope, a really simple little microscope. This is it. This is a Leeuwenhoek's microscope. It's got a tiny little lens there, the size of a drop of water. Really small, but he was a really good lens maker, and that little lens was really cool. And he'd hold this thing up, he'd put a specimen on that drop, and then he could focus it by turning the screw, and he'd put his eye right to it. And with that simple microscope, one lens microscope, this guy was the father of microbiology. He found protozoa, bacteria, red blood cells. He looked at his blood, it was made of cells, and he looked at his sperm, and it was made of spermatozoa. Can you imagine? First time seeing sperm? That must have really been a trip for him. I mean, really. So so I got the bug for doing for microscopes. This is me a long time ago as a PhD student, and I started off really after college as a photographer and a filmmaker. So I got the chance to start making films through the microscope. I thought, this is cool. Films and microscopes, that really goes well together. Now, this was my normal camera back here as a Bolex camera, but I got a chance to do some of the special effects in the first Superman film, and they lent me a Panavision camera that I put on attached to the wall, and we did photographs of human pancreatic cancer cells to make it look like the surface of Krypton. And it actually worked, and it was one of the first shots in the movie. So I've been playing with microscopes for a long time. A really long time, and I've never ceased to be amazed at what I see. It's just completely incredible. So let me show you a little bit about the space that I'm talking about now. This is a meter, a tenth of a meter, centimeter is a hundredth of a meter, millimeter is a thousandth of a meter, and so forth. There's a micron a micron is a millionth of a meter, and then down to a nanometer, which is a billionth of a meter. It gets smaller and smaller down to an angstrom unit, which is the diameter of a single hydrogen atom. That's how small that is. So the smallest thing we can see is about, in this range here, is about a tenth of a millimeter. That's the size of a human hair. The size of a human hair is about a tenth of a millimeter. That's about the smallest thing we can see. And our cells in our body, of which we have hundreds of trillions of cells, each cell is about 10 times smaller than that. Each cell is about 10 micrometers. That means about 10 human cells can fit across the diameter of that human hair. So cells are really small. You can't see them without a microscope, and you have to have a fairly good microscope to see them. This is a cell of the immune system called mast cells, MAST. They cause inflammation and allergy. Those are cells that I studied for many, many years. And I studied the immune system and how these cells interact with one another. So this is a cell called the macrophage. Now, do I click on this to make this work? Let's see. Let me back this up. So there's this macrophage, and what it does, it's looking for anything foreign in the body, bacteria, stuff we breathe in, and it travels all across our cells looking for something foreign, and when it finds it, it grabs it, eats it, and digests it. So here's a little bit of debris here, and that macrophage is going to grab it, it recognizes it as foreign, it ingests it, and it digests it. So there's billions of these cells all the time taking care of our bodies, running around looking for anything foreign and destroying them. It's an incredible search and destroy mission. Now let's see what I'm doing here. Got to figure out. JD or okay. There we go. So this is the nerve cell. Now I took all these pictures when I was a oops. I took these movies when I was a young graduate student, and these are what nerve cells look like. You know, the cells in our brains, they're not just static. They're always moving around looking for new connections. We're always trying to find new neural networks, and the tips of these nerve cells are always finding new connections and making new connections. When we learn things like we're learning tonight, these nerve cells are going around finding new connections and breaking old connections sometimes, depending on what's happening. So that's what our nerve cells are doing. They're doing that all the time. It's pretty amazing. These are time. That was about 25 times. Those are both 20 time lapse, 25 times normal speed. Now here's another one. This is heart cells. And if you look at heart cells, they're quite amazing. They just sit there beating. If you this is from a rat. You take this is like 5 heart cells. If you take them out of the rat and put them in a dish with some medium, they just sit there and beat because that's their job. And the DNA that they're making, they're expressing out of their nucleus, makes the cardiac protein muscles that make the beating happen. So it's in the cards, it's in the DNA, they just do it. Now nerve cells hook up to them also and control that beating, but they'll beat on their own without any nerve cell intervention. It's pretty amazing. They're very tenacious, heart cells, to say the least. Let's see. Okay. They're bathed in glucose and you know, calcium, all the sorts of things that they need. They're in a solution and kept at room temperature, and they'll last for quite a while. Yeah, yeah. So I've been got really interested in making microscopes, not only looking through microscopes but making them. So I started inventing microscopes about a little over 20 years ago. And the reason I started inventing microscopes is because I needed to see, from the research I was doing, to see things in 3D. Microscopes had traditionally been 2D microscopes, and I needed to see things in 3D with good depth of field, and they didn't have microscopes that would do that. So I started working with different types of lighting to get oblique lighting to look at things from an angle, and tilted angles, and getting 2 tilted angles at the same time, and being able to start seeing 3D. This was the first microscope that I made in Santa Monica, 1992 and 93. This is my new microscope, new prototype, that I just finished a month ago. And this microscope is really quite amazing. It's all automated. It's digital 3D microscope. And it's run by a computer, and it does everything automatically and in 3D, and it's super cool. And hopefully one of these days there's going to be a lot of these things around somewhere in different people's labs. So let me show you. I'm going to get out of the way here if I can. What happens in a microscope? You only get a little depth of field. So you see that's some moon dust there, and that's what you see in the microscope. But as you focus through, you get different focus levels, but you can't really see what's happening. But these microscopes that I did, that puts it all together, and now you see it as one quite amazing little grain of sand. And you'll notice one thing about this grain of sand: there's a hole in the middle, and that was caused by a micrometeorite. A micrometeorite. There's a grain of moon sand. It hit this little group of grains of sand at about 20,000 miles an hour, and it caused this little hole. And you see it's kind of melted. It's all melted with kind of a glassy envelope, and that's called an agglutinate particle. And the moon is filled with this because there's no atmosphere on the moon. There's no water. So when you see grains of sand, they look very different than sand on Earth. And I'm going to show you some of this in a bit. And they would look different because there's no atmosphere and there's no water. So is that really what that grain of sand looks like? What I want to point out in this talk is there's no such thing as what things really look like. You only look at something from a point of view. So here's a grain of sand from the moon, an agglutinate particle, with a light microscope. Here's the same exact grain of sand from the same exact angle with a scanning electron microscope. And here's the same thing again with an X-ray microscope. So this one sees right through the middle of it, and it sees what's in the inside. This scanning electron microscope sees all the details of the surface, and the light microscope can see how translucent something is, whether it's opaque, whether it's got a polarized light or not, whether it has color. So every technique, every angle that we look at the world from gives you more information. There's no such thing as seeing things as they really truly are. So what I want to show you now is just a range of ordinary things. Because one of the things I have a lot of fun doing is just taking ordinary things and seeing what it looks like in a microscope. So you have a hard time believing it, but you know what this is? This is like a non-abrasive Scotch-Brite scrubber that you scrub your dishes with. And it's non-abrasive because that blue side that's kind of plasticky kind of, they're made of these little nubs of plastic, so they're non-abrasive. If you get the abrasive one, it looks like it's sandpaper. And so these are the ones used for Teflon and so forth. But it looks kind of like a neural network. It's pretty cool looking. And you'd never guess that that's what that is. This is Lipton tea, which I was pretty surprised that it looked like this actually, because I've seen leaves before. Now this I thought looked a little bit like outer space. It's Colgate toothpaste. And it's got little bits of stuff in it to be abrasive for your teeth. This one here is crystallized sugar. So there's if you put sugar and warm water, and put a lot of sugar in and let it cool off, it'll start to crystallize. The sugar crystals will come out of solution, and sugar will come out of solution and crystallize. And this is what it looks like. This is polarized light. So the color here, the color here is from polarized light. Polarized light is light that, light, if light's going toward you like this, it's vibrating this way, it's vibrating this way, all 360 degrees, all different angles vibrating as it's coming toward you. Polarized light is light that vibrates at a particular direction. So if you allow only polarized light to go, they have what are called polarizing filters, that'll cut out all the light except light going in this direction, let's say. And then you cross it with another polarized filter that blocks all light except that goes this direction. What you get is blackness. You get no light coming through. But if something's in the middle, a crystal, it twists the polarization of the light. The amount it twists it gives a different color. So it's that twisting of the polarized light that gives these colors here. And they're pretty amazing. Now here's another example of wine. This is Merlot wine. And at the end of the night or in the morning, you come and you see a little drop of the wine in your glass, and it looks like it's all chunky and weird at the bottom. Those are wine crystals. And every type of wine has a different shaped crystal. This is from Merlot wine, from Merlot. And they're all completely amazing different shapes in polarized light again. So I want to talk a little bit about flowers now, because when I moved to the island 8 years ago, I got completely fascinated by hibiscus flowers because they're so beautiful. They're such a trip. They're really great. And I started taking a lot of pictures of hibiscus flowers, thousands and thousands of pictures, through the microscope, through my close-up lens and through the microscope. And I began to think, how does a little bug like this little ant here, how does that little ant see a flower? It certainly sees the flower differently than we do. First of all, it sees it really close up. It sees it like a microscope, because this thing here is that big to it. That's the stigma of that flower there. So that's the stigma of the flower. That's probably what it looks like to that ant. That's how big it would be. And that's the receptive part of the female organ of flowers. This is where the part where that receives the pollen, and then the plant can become pollinated, and I'll show you a little more on that. So here is the male part of the plant. When you see the hibiscus, you see all the little yellow dots. Well, the yellow dots are made of, this you'd think is one yellow dot, but it's made of hundreds of individual grains of pollen. And each of these grains of pollen, every one of them, has 2 sperm cells. So that's what they are. They got 2 sperm cells. And what happens is I'll go to that in a minute. Let's go back and look for a second. So you see there's like hundreds of pollen grains in here, and this is the female part. I want to show you what it looks like right in here. If you just look right in that area here, these are called the filaments that hold up the, let's take a look at that in a microscope. That's what it looks like. These are the cells. These are the individual cells that make up that little tiny bit of that flower. And look at this. You see this stuff here? It's unbelievable. That's nectar. That's sweet nectar that the plant produces to attract the bees and the insects. So that's why, you know, if you ever look at hibiscus flowers, they're filled with insects. If you take them apart, you're going to see tons of insects there because they live in there. That's one of them. This is one of the little strange guys who lives in it's tiny. There's a really, really small little bug. But look, it's like a little Willy Wonka land for this guy. It's like his candy land, completely in candy land. So that's why they live there. I have, yeah. And in 3D. Not tonight. But in 3D, no, I didn't bring any of that. But I've done it in 3D as well. So here's the deal. The idea is as these little birds, as these little bees and are moving around, the idea is either a bee or one of these little insects rubs up against the pollen. Now the pollen is really sticky. It's really waxy and super sticky, and it sticks to everything. So the idea is these little bugs go around and hopefully deposit a single grain of pollen on the stigma of that plant. And what will happen is this has now been received by the stigma. It'll travel into the middle of the stigma. It's quite amazing. And what it'll do, there it is, there's that little bit of pollen right there. It'll travel all the way down this style. I've cut this open here. And into something called a pollen tube. There's a pollen tube down the middle of this thing. The pollen goes all the way down. I'm going to show you what that looks closer up here. And what that is there is the ovary and the ovules, and there's the pollen tube. So that pollen traveled all the way down that distance until it got here, and then it pollinated those seeds, or those ovules, which then become seeds. Once the sperm cell has combined with the ovules, they become seeds, and this ovary becomes the fruit. So if this were, for example, a flower of an orange, then these would be the seeds of the orange, and that would become the orange. Well, that's the birds and the bees. And that's a seed. That's the beginning of a seed just forming after it's been pollinated by some pollen. That's in a cucumber. So it's an amazing world there. And I want to talk about another amazing world that I've been involved with. When I got here, I started taking a lot of pictures of sand. And sand is kind of ordinary, but sand is much more surprising than we can imagine. It's incredibly beautiful, and sand is a snapshot in time. Sand is not a fixed thing. It's coming from somewhere, and it's going somewhere. In the case of sand from mineral sources, it's being chipped off, or I should say eroded off of rock into little, and that's being transported down to rivers, onto beaches, until they finally erode away into mud and silt and clay, into nothing. So it takes long periods of time. So when we just look at sand, we're looking at a snapshot in time. We're not seeing where it's coming from and where it's going to. But sand is really quite extraordinary. So this is what most sand looks like. It's made of quartz crystal. So this is sand from Florida. And quartz crystal is the quartz and feldspar are the erosion products of granite. So most of the continents are made of granite, and they erode into sand, little bits. In fact, if you look at of granite through a microscope, a slice of granite through a microscope, you see the little grains. And they're about the size of grains of sand. And they get liberated through temperature, ice, wind, water. And these little grains get liberated one at a time, go down the river, and they form the sand. So most sand is this kind of sand here. But a lot of sand is quite amazingly beautiful. This is sand from Big Sur. And there's pink quartz in it, rose quartz, and some really beautiful other things. Here's some sand from Namibia in Africa, and it's just all jewels. So this is taken from a downstream from a jewel mine in Africa. And if you look at the sand, it's just filled with jewels. It's completely amazing. So mineral sands are really quite incredible. But we have different kind of sands here on Hawaii. We have basalt. First of all, our mineralogy here, or we have the mineralogy from the bottom of the ocean, which is basalt. Basalt is heavier. The continents are higher and they're lighter, so it's mainly mainly on islands, it's basalt. So we have these basaltic lava flows. Many of us have been to the beach here in Hana. That's a red sand beach in Hana. This is what the sand looks like in a microscope from that beach. Pretty beautiful. And we also have in Hawaii, in the southern tip of Hawaii, a green sand beach. This is at South Point in Hawaii. And yeah. Yeah. This is the beach at South. It's called South Point. Yeah, it's called South Point, and it's fairly hard to get to. But the beach is solid olivine, the whole thing, and it glows, it glistens bright green. It's completely amazing. And if you look at the sand through a microscope, that's what it looks like. It's solid olivine, all eroded from the local mountains right there. It's an amazing, amazing thing. So we have some pretty incredible sand on the Hawaiian Islands. Now this is some sand from McKenna Beach. Now first thing you notice, almost all of it is highly eroded bits of shell fragments. Because on islands, a lot of the sand is from shell. It's not from quartz crystal, because we don't have quartz crystal here. A lot of it's from shell. But I want to zoom into the middle of this and show you that it's a lot more than just eroded shell. So now in the middle here, we're seeing some other stuff coming out. And if we get a little closer in, look what's in there. This is from the volcano. These two things here are sea urchin spines. This is a little tube worm. And this here is a bit of a foraminifera, which is a type of a protist or a protozoa that makes a little house for itself. So the sand, this is a little bit of sea urchin spine as well. Sand has some amazing stuff in it from Maui, really, really quite incredible. And here's some examples of sand from Lahaina, from a beach in Lahaina. Look, there's a little thing called a sponge spicule. It's nature's first light guide. So what this is, these sponge spicules live in sponges, and sponges have symbiotic algae that live inside. But these sponge spicules bring light into the inside of the sponge so that these algae can photosynthesize and do their job. So it's one of nature's first light guides that was, developed about 300 or 400 million years ago. Little shell fragments, little bits of coral. This is a foraminifera. This is a sea urchin spine. There's some coral. So there's just some amazing stuff in sand locally what we have here. Really quite amazing. Some beaches are completely made of these foraminifera called forams. And this is one from Okinawa. And all the grains of sand on the beach look like little tiny stars. And they're the little houses of these little foraminifera. Look like little stars with little studded pearls on them. Completely amazing. You need to look at it in a microscope to see it. So I want to go from stars here to outer space. Sand is also ubiquitous in outer space. Space is filled with sand. We call them micrometeorites, and they're everywhere. And some of these come together, and they make asteroids and moons and planets as they accrete together. But these were found in the South Pole. These survived the journey in, and these are really, really, really tiny. They're like if you take a very fine pencil and put a dot on a piece of paper, that's about how large these would be. Very, very small and very beautiful. And space is filled with this, and this is sort of the stuff that our universe is made of. Well, Joe and I have had the privilege to get some sand from the moon. Joe Ritter and I have a grant from NASA to look at moon sand. And so we've been looking at moon sand, and moon sand's quite amazing because it's made of the same stuff the Earth is made of, but it looks so different because it's weathered so differently. So this is this is the first picture of moon sand. I thought it looked like the moon. I thought it was kind of cool looking. But if you look at sand just kind of casually from the moon, this is what it looks like. It doesn't look like there's much there. When you start to look more closely and go through it, there's some pretty amazing stuff. So a lot of it is this basaltic type minerals, but then you have a lot of minerals like anorthosite and plagioclase and other kinds of minerals that we have on Earth. And again, a lot of what happens on the moon is there's been so many bombardments. The moon has been bombarded for over 4 billion years by micrometeorites, meteorites, asteroids, all kinds of stuff has bombarded the moon. And all these bombardments sort of break the sand up and smash it back together again. So here's a grain of sand that is a glutinate particle. It's made of all these particles that have been fused together by these impacts. Now look at, again, how different it looks. This is a light microscope, scanning electron microscope, X-ray microscope. You get a different view each time of what you see. A lot of these impacts cause these different minerals to fuse together, a metamorphic rock. So here we have, like, you know, little bits of different rock that have been fused together by these incredible impacts on the moon. And some of them are really beautiful. Also on the moon, there's a lot of glassy spherules. And the glassy spherules, a lot of them come because about 3.5 billion years ago, there were volcanoes on the moon. And they had these fire fountain volcanoes, and they would spew out these microscopic drops of glass in all different colors, and some of them are completely amazing. And they're still there. Some of them get blown up. Here's one of these pieces of glass that was formed, and as it was spinning around, it was just starting to come apart like a dumbbell when it froze in time. And when things freeze in time on the moon, they stay that way. It doesn't get eroded away. The footprints on the moon are still there, you know, just as they were to be 50 years ago. Some of this stuff is really beautiful green glass. It is from these fire fountain volcanoes. Some of it is iron and iron oxide, red ones, which are really beautiful. There's a lot of colored glass, and the colored glass keeps getting broken up and reformed but through these impacts. So the moon sand is really much more beautiful than one would expect it to be. Here's one of my favorite little tiny grains of sand from the moon. It's about 4 billion years old, and you can still see the entire crystal structure of this grain of sand. It's really, really quite something. Now, I want to end up, I think. So we've been looking in kind of this area here of how small things are, but actually we have microscopes that will go all the way down to an angstrom. They can actually see atoms. So what I want to do, and this is a picture of atoms. These are some cobalt atoms with a scanning probe microscope. And I want to end up by showing you a little movie done by some French researchers. Now, let me see what I have to do here. Whoop. What happened? I got to blow this up. Hold on. What did I do? I'm going to try this again. Oh. No, right. Okay, maximize it. There we go. Okay.
Bubbles of gas trapped when the metal is molded. In bursting, they leave holes that form irregular oblong shapes, characteristic of aluminium particles.
Louder.
Around the cavities, these aluminium particles. These cavities are the ends of two of these particles. Drawing closer, they appear like huge mounds. The surface streaks are the result of polishing, fine scratches magnified 1,500 times. The tiny pebbles glued to the surface of the aluminium are probably particles of silicon. Silicon and magnesium are added to reinforce the metal. Without them, the support would be as fragile as the aluminium foil used in a kitchen. Closer still, and a contrasting pattern of black and white emerges. We're now looking inside the alloy itself. The white is pure aluminium. The black, an indiscernible mixture, is made up of faults in the material and also precipitations of magnesium and silicon. Although they play a fundamental role in the strengthening of the alloy, it's not known how they occur. Research is still being done on it.
Now you're going to notice we get to about 1 or 2 million times, the whole universe changes.
And here is the actual atomic structure of the aluminium. Ordered columns of atoms forming a checkered pattern, stacked in all directions.
So those are the individual atoms that make up the aluminum. So this is a couple angstroms in diameter.
Beyond this, nothing more is visible for the moment.
We need our younger people to figure that one out. So, thank you very much. I better stop this. Hold on. Let me stop this. Okay. There we go. Thank you. Appreciate it. So I don't know, are there any questions that anybody has? I might be able to answer them.
Gary, how do you organize the grains of moon sand?
To get them all to look the same, you mean?
Or no, how do you rotate?
I use acupuncture needles to move them around. That's the best thing I've found so far is the acupuncture needles. It's manual manipulation and a lot of trial and error. You get good at it after a while. I actually used to put in biology. I used to put things together that were really small biological things and have them grow, and so I have pretty good dexterity on that level. Yeah. That's a good question, and if I knew more about geology, I could really give you a good answer, and I'm a little challenged there. So the question is, what minerals are there and what uses do they have? Is that correct? So on Earth, aluminum is one of the silicates, and aluminum are one of the most prevalent things on Earth. And the stuff like gold and silver and uranium, there's very, very little of that kind of stuff. Yeah.
You said at one stage that birds can see magnetic fields. Do you mean that, or do you mean they have a sensory awareness of magnetic fields?
They have a sensory awareness of it. However, it happens in their eye. Yes, the apparatus, it appears the apparatus is in their eye, and it also appears that biology or evolution has somehow cashed in on the entanglement of electrons, because as they understand now, and this is not fully understood, but they think that these cells in the eye produce entangled electrons that go in two different directions, and somehow those get received by these receiver cells, and that tells you, because one of them goes through the magnetic field in one direction and the other in another direction, it gives information about the magnetic field. But it's not understood, but it looks like they've actually capitalized on the idea of entanglement in quantum physics. It's pretty amazing.
There's some recent research that is indicating that they believe that humans have the same ability for the sixth sense, being able to perceive magnetic.
The magnetic field. Interesting.
In that video with the aluminum seat, could you say that was going from a point of view to no point of view?
I would say it was continuously changing point of view, and in that journey there was a lot to see. So it wasn't just a bicycle stem. It was an entire journey through all the way down to a single atom. So it was a journey of different points of view, is what I would call it.
What is your feeling about the ability, not with the eye, but the human ability to perceive from no point of view? Because the eye itself has the ability to perceive and respond to a single photon. So the human being has some abilities that we haven't tapped into. And I'm just wondering, as you sit in there, everything is from a point of view. And I believe that's the case in terms of normal vision. But there's other ways of perceiving. I'm just wondering, that video made me...
Well, people here who are meditators, attempt to put themselves in a pointless point of view or some point of view that isn't the point of view. I'm not sure that's actually possible, but it seems like it is. I know because I meditate, and it seems like you're in no point of view. But in fact, I'm a human being with all my heart is beating and I'm breathing and all this stuff is happening. So my assumption is I'm seeing that nothingness from a human point of view. And maybe a dog would see, dogs are really good meditators. Maybe they see it from a different point of view. Yeah.
Beyond your 3D microscope, what do you see in the way of future microscope development in terms of being able to see the world as we can now?
I think in terms of magnification, we've done really well. Because we've got down to seeing where an atom is and how big it is. To see inside that atom is probably not going to be visualized in the sense that we think we can visualize, because they're made of subatomic energy, particles of energy and fields and stuff that I'm not sure that we're going to see it in the way. We can understand it and see it in drawings, and we can understand these subatomic particles in theoretical ways. I'm not sure microscopes will be able to do that, but I could be wrong. But I mean, who would have thought you could see an atom with a microscope 20 years ago, 15 years ago? So, things changed. Yeah, Steph?
Yes, Gary, there's a Roxy Sweetwater in Second Life, and she says, Nixie from Maui would like to know more about your new book, Floratica.
Floratica. So Floratica is a book that I did when I got very turned on to flowers, as I said, and I took thousands of pictures of flowers, and some of them were really pretty sensual. So I got all my most erotic and sensual pictures together and put them into a book called Floratica. Yeah.
We tend to think of infinity in terms of outer space and, as far as you could go, and it's infinite, whatever. But isn't the same true going smaller?
I think
Limit to how small something?
there does appear to be a limit, and it's called the Planck constant and Planck limit, about 10 to the minus 34. Is that right? It's something like that. At that point, things. Well, Joe, how would you describe the Planck limit?
Well, so the question is, what is the smallest continuous particle, right? And so that's a function of energy, right? And you have
Planck length is how long it takes for lights to go in the Planck time. In 1 Planck unit of Planck time.
I yield to my colleague.
I don't quite remember how they worked it out, but I'm pretty sure there's a good number of videos that you can find online.
So when things get really small, Garrett maybe have a better answer to this than I do. Garrett, what do you say?
Basically, you combine the gravitational constant and Planck's constant to scale of quantum mechanics. That gives you a literal spin probe without creating a black hole that won't let you measure any closer.
So that's 10 to the minus 34. Okay, so 10 to the minus 34, to give you an idea how small that is. Right, and we maybe never will. Is that sort of like the uncertainty principle? Is the uncertainty principle? Right, theoretically, but right. Yeah. So that means 0.34 zeros. followed by a 1. That's a really small number.
Do we know if the composition of the dust or the sand on Mars is similar to
well, Mars is different than the Moon. The Moon and Earth are the same because the Moon, in fact, when we went to the Moon, we realized that the Moon was formed probably about 4.6 million years ago by an impact. In those days, billions of years ago, there was a lot of stuff flying around the solar system, and there wasn't only the planets that we see today. And Earth got impacted by about a Mars-sized object, a big object that slammed into it, and the Earth sort of, a lot of it went into the Earth, and a lot of it spun off into debris that became the Moon. And that's why the minerals and isotopes of minerals on Earth are exactly the same they are on the Moon, because it was made of the same stuff. Now, when you go to look at other planets and other asteroids and other moons, they have different signatures. So yes, Mars would look different, and they're starting. Curiosity mission is now just beginning to look close up at the geology of Mars, and hopefully we're going to get some good answers to that soon. That's an excellent question. I'd like to know the answer to that one too. Why is there not one of my microscopes on Curiosity on Mars? And in fact, Joe and I are working on trying to get one of our microscopes into outer space, onto Skylab. Because Skylab does a lot of stuff with microscopes in outer space. So, so the space station, sorry, on the space station. Yeah. Yes.
The thing with the when they're looking at the surface of Mars with the dust, they've actually found iron, and they're not entirely sure. It took on a bit of a strange shape, and they're not entirely sure. They're trying to figure out the answer right now.
Good. See, this is all really happening right now because we just, landed on Mars not that long ago, and stuff happens slowly there. Stuff doesn't happen real fast, but we're going to get answers to this stuff. There'll be papers written about them over the next year or two about what is happening. Right. Where they were drilling into rock.
They were successfully drilling, and now they've run a lot of samples through Curiosity to kind of clean out anything that was in there, and now just stand by for the next analysis.
Exactly. So they're yeah, basically stay tuned because a lot of that information is going to come to us over the next year or so, and it'll be published in magazines. Okay, well, Garrett's got to field that one. Garrett, what's the deal? Yeah. I know I have not physically seen it myself, but it looks super cool. Do you have one?
I've read the guy's thesis, and we've talked. I would love to have one of those. It's an interesting device, and a lot of other uses aside from the just change focus stuff. Because the whole light field, it's got a whole bunch of little apertures. I have the thesis if you were interested in reading it.
I don't know if anybody's used it with the 3D microscope. Use it together. It seems like it would be a natural.
I think so. I think we should get a hold of this guy. Yeah, I think we should get a hold of him and see what we can work together on. I think that's a really good idea.
There's a new probe that went out, I read, named after the planet, that has determined that the Big Bang occurred 80 million years before.
Yeah, it seems to be older, 800 million years. So instead of 13.7 billion years, it looks like it's maybe almost 13.8 billion years. 13.78. That's 8.2. Sorry.
The interesting thing is the asymmetry part.
No, tell me about this.
If you look at the whole image, on the right side of it, it's very slightly brighter than on the left side.
This is of the Big Bang image
Of the whole universe.
of the early universe.
There's an asymmetry on the scale of the whole universe, and that's absolutely what Garrett said.
And it seems to be much clumpier than we thought it would be as well. Is that right, Garrett? That it's much clumpier than we thought it was going to be.
I have a tab open on my browser.
Ask Garrett how he responds to the asymmetry.
Gary, what do you have to say about the asymmetry in that picture?
One of the most amazing things, I mean, we're used to general relativity thinking that there's no such thing as a fixed frame of reference in our universe, but in fact there is, because the Big Bang itself happened everywhere but uniformly. So we can actually look and see radiation from different locations and tell what stationary is within our universe based on the radiation coming in from at the same Doppler shift from all directions. So if you see one shift in the Planck satellite, looking at the cosmic background radiation, that could be just they haven't filtered out to get a completely stationary point with respect to the cosmic background. But I haven't looked at it yet in depth.
So they're saying that's not what they're saying that's not what they're talking about.
They're talking about some abnormalities in other modes.
Yeah.
Okay. I don't know. It's really interesting, and it's probably one of the best views into the evolution of our universe from the Big Bang. There's some amazing things to be found about cosmology and inflation all the way back to back when and how it all started. So there's a lot of fascinating stuff there, and yeah, cosmology is definitely something to keep your eye on.
Yeah, well, stay tuned. Yes, young man?
I have a bit of a question that I've been having trouble understanding. The visible universe is finite, but what's considered the whole universe is actually considered infinite, which I've always found a bit confusing since the universe itself is finite in time. So, how I'm a bit confused on how the universe itself could be infinite while it's only existed for a finite amount of time.
I don't think it's supposed to be infinite. I think what's visible is what you can see within what is called a light cone, right? And we can talk about this afterwards in some microscopy. But I think the short answer is it's believed that there was a time, and Garrett knows all about this, but there's an inflaton in the universe actually expanded exponentially in size. And although things aren't supposed to go speed of light or faster than the speed of light if they have mass, space can expand faster than the speed of light. So it's bigger than what's observable. I'd be happy to talk to you about cosmology afterwards. I think there's time for just a couple more questions for Gary before we let everybody go. But
can we see dark matter? We talked about different frequencies of UV.
We can see its effect. We can't literally see it. We see its effect on other matter. That's if you believe in it? Is that right? Really? It has a gravitational effect on other matter. Yeah. Yes.
Talking about redshift, finally I was getting somewhere. There are surely more than one reason. There is more than one reason for a redshift. It's always assumed it's a Doppler effect. Could it not be? And this is where I can get thrown out for being too wacky. Could it not be? Given we're thinking about dark matter, dark energy. Could it not be that all this light is passing through a medium that accelerates and retards the velocity of light, which would give the same effect? I think it's I couldn't work it out. I think it's if it slows light as it travels, does that not have the same effect as something shooting away from us? And I always love having a go at the Big Bang.
Well, when Hubble came up with this idea, it was not readily accepted at all. He had to do a lot of convincing to a lot of people because they had all kinds of reasons why it could be something else. However, over the years, it appears there's an awful lot of strong evidence that's saying the redshift is cosmological in nature.
I understand that, but I'm saying that we are assuming, I understand it's cosmological, but the cosmological acceptance is that everything is expanding. Could you not get the same effect if we were to reinvent phlogiston or whatever it was that existed?
There's some ether in the media, well, that was one of the ideas then. But that seemed the ether has not really been found yet, has it?
there is some effective refractive index within a galactic medium too.
Because what we have now is extremely precise models of the evolution of the cosmos on the intergalactic scale, which show you clumps of galaxies and precisely how these evolved from the Big Bang. So you don't have to, I mean, they're all this intricate web of interconnections that go into the standard cosmological model, which really leave a lot of
Well,
Art and those people toil that they still publish papers suggesting it?
Yes. Yes, they do.
well, with that, that's the great thing about science. It's a never-ending argument. That's what it's all about. That's what it's all about.
I always thought that birds had six color receptors.
As far as I know, there's 4. But we can, maybe some birds do, but
Humans actually have another 4th one too, but it's not on the graphs you have, but it's different and it affects dilation. But yes, birds have extra. And in fact, taste is interesting in other animals too, different numbers of taste buds. But that's
yes.
I think we all could probably talk Gary for hours. I know I like to. Why don't we stop here, but you can go up and see him, and let's thank our guests tonight, Gary. It's all yours.
Thank you. Thank you very much. Thank you. Thank you. Thank you. Thank you. Thank you very much. Thank you. I could talk for a long time. I could definitely talk for a long time. Thank you. Thanks for coming. You've heard my story so many times. Thank you. Thank you. We're working on it. It's about half gone.